everyone, welcome to Biomagic. Very important topic for class 12th, exam oriented, that is lac operon. Lac operon. Now, lac operon, before going to that, in prokaryotes, Jacob and Monod expressed the characteristics of genes, how they are expressing, right? The genes in a cell are expressed to perform a particular function or a set of function. This is given by Jacob and Monod. To explain that, they gave both induction and repression. Okay, so here I am going to deal with lac operon, that is an induction type of gene expression in prokaryotes, especially operon concept. Now, what is operon? Operon is a set of genes which consist of structural gene, regulator gene, promoter gene, and that set is known as operon. Okay, that we will come to know just now, right? Now, for example, sugar lactose. Lactose is added to the culture of E. coli, then suddenly there will be induction of three enzymes. That means when we are adding lactose, there will be the production of three enzymes. That means activation of three enzymes. And what they will do, they are necessary for the breakdown of lactose into glucose and galactose. Right? So, lactose is added, three enzymes are active and they are necessary for conversion of lactose into glucose and galactose. Now, which are the three enzymes? Beta galactosidase, permease, transacetylase. Now, beta galactosidase is regulated by the gene LAC-Z. Permease regulated by LAC-Y. Transacetylase regulated by LAC-A. LAC is lactose, right? So these three genes are necessary for the production of these three enzymes. Now, beta galactosidase is essential for the breakdown of lactose into glucose and galactose. These two, permease is permitting the beta galactosidase inside the cell, and this is also enhancing the functioning of beta galactosidase. So three of them are necessary for the breakdown of breakdown of lactose. So these three are known as structural genes. So operon concept, structural gene, right? Little concept. Each gene segment is known as cistron. Small gene segment is known as cistron. Each gene segment. Messenger RNA, which is formed as a result of transcription, covering all cistrons, that means all segments of genes, is known as polycistronic. That means a number of segments, right? Three genes are regulated, and it is regulated by the operator gene. So we have studied structural gene, operator gene, and a number of segment of genes. These units are called operon. Structural gene, regulator gene, operator gene, right? Now, regulator gene is producing a protein known as repressor, which will bind to the operator and switch off or turned off the functioning of RNA polymerase. So, if RNA polymerase is not functioning, it will not make active the enzymes. So, enzymes will not produce the functioning or the substances which is need for the breakdown of lactose. Now, suppose it is induction. So, we are providing lactose in the medium. If lactose is present in the medium, it will bind to the operator, and there the transcription starts. And it is what operon induction. Now, let's see with the help of diagram. Now, this is what a lac operon starting. This is regulator gene, promoter gene, operator gene, and these three are the structural gene: LAC-Z, LAC-Y, lac A. These three are the structural gene. Lac Z is responsible for the production of beta galactosidase. Lac Y is producing permease. Lac A is producing trans-acetylase. Right? So these three are the structural gene. Now, regulator gene, as I told you, it is transcription happening, so production of mRNA, which is producing a proton repressor. Right now, repressor will go and bind with the operator. Repressor binds with the operator. So from here, the promoter, the RNA polymerase, will not be able to go through it and make these genes to produce these enzymes, because the repressor, after binding, blocking the area, so RNA polymerase cannot go and make the gene transcribe, right? So there is no production of these three enzymes, so there will be no breakdown of lactose, right? Now, for example, I am giving lactose here, lactose in the medium I am giving. This lactose, what will do, it will bind to the repressor. For example, this is the repressor, this is the lactose, so it will go and bind the repressor, right? So this is what happens, binding of lactose with the repressor. So repressor will not able to go to the operator, so this will not happen, and this will not block the pathway of RNA polymerase. So transcription happens, production of three enzymes, and this will help in the breakdown of lactose into glucose and galactose. Lactose into glucose and galactose. This is what induction, that means after adding lactose, we are inducing the proper functioning of these three enzymes for the breakdown of lactose. Hope you understood. Put your comments and like, share, subscribe. Also press the bell icon so that whenever I am giving any video, you will get it, right? These are all exam oriented portions. Thank you so much.